Hello, and welcome to another episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea for Halteris Associates. Today, I'd like to talk about malaria. Malaria has been in the news recently. There have been a few cases in Florida and a couple other places. I'll talk about that a little bit. But I want to talk about the overall picture of malaria. And I'm going to start by talking about what malaria is, some of the history of malaria, and, of course, the diagnostics. As you probably know, malaria is a mosquito-borne disease, and it infects both humans and also a number of other vertebrates, including birds. It's caused by a group of parasites called the Plasmodium genus. Talk more about the various species in a few minutes. And it's transmitted by the genus Anopheles of mosquitoes. The mosquitoes actually have the organism growing within its saliva. It's transmitted through the saliva. And once the mosquito bites someone, usually the symptoms begin about 10 to 15 days. Symptoms include shivering, fatigue, fever, vomiting, headaches, joint pain, potentially even jaundice, and occasionally coma, and often death. Malaria symptoms can recur in a couple of ways. One is through what's called recrudescence, which is where the infection was never actually eliminated through drug treatment or any other form of intervention, and the symptoms come back again. Another is from recurrence, and this is in a situation where there are hidden forms of the infection. I'm going to talk about that a little bit later. And the third is actual reinfection, where an individual had a disease, it was in fact cured, but they are in an endemic area, so it recurs again through a new infection. One of the major features of malaria that differentiates it from so many other diseases is a recurrence of fever that can occur. And that can be every 2 to 4 days, depending on the particular species of Plasmodium that causes the infection. There are 5 major species of Plasmodium associated with malaria. The first is Plasmodium falciparum. That causes about 75% of cases worldwide. It is also by far the most deadly of the species. The second most common is Plasmodium vivax. That accounts for about 20% of the additional infections. And then the other 3 are Ovale, Malariae, and Knowlesi. They cause the additional 5% or so of infections, and so are not very prevalent. The WHO estimated in 2021 that there were approximately 247 million cases of malaria worldwide, quite a large number, with about 619,000 deaths. And of those, 67% were in children less than 5, so they are particularly susceptible. Malaria has been eliminated in the European region only since about 2015, so not very long ago. In contrast, in the United States, malaria was eliminated in 1951, but it has recurred upon occasion. For instance, in 2003, there were 8 cases in Florida. And then most recently, here in 2023, we had another 4 cases in Florida, 1 in Texas and 1 in Maryland. So occasionally some of these infections do occur even today. Now, there are 2 types of infection. One would be where someone traveled and brought malaria back home with them. I'm not talking about that in this case. I'm talking about people that were actually infected in the United States by mosquitoes in the United States. So it's rare, but it still occurs. Most places with endemic malaria are in the equatorial belt. And so you'll see countries in South America and Central America, Africa in particular, and Asia. And there are today 84 endemic countries throughout the world. Unfortunately, sub-Saharan Africa harbors most of the deaths, approximately 85 to 90% worldwide. And most all those are due to P. falciparum. Malaria is an ancient disease, probably 50 to 100,000 years old in the human and primate population. It seemed to have been fairly confined until about 10,000 years ago with the dawn of the agricultural age, when it began to spread broadly. There are many historical references to malaria in documents throughout history, and that's mostly because of the occurrence of these periodic fevers, which is very peculiar to malaria. For instance, Hippocrates noted that there were 2 types of fever that he saw. One he called tertian, which was an every 3-day fever, or quartan, which is every 4-day fever. So very, very characteristic of malaria. In fact, those terms are still used today based on Hippocrates' writings. The Roman author Lucius Columella actually believed that this was caused by insects from swamps, which was a very interesting impression based on his observations within the Roman Empire. In fact, malaria, which wasn't yet called that, in the Roman Empire was so common that they called it the Roman fever. Later in history, the thing that we now call malaria was called ague. It was called marsh fever. And it wasn't until the Middle Ages when the Italians named it malaria, or bad air. And of course, this is before germ theory. So they had the notion that bad air in some form or another, coming from marshes and swamps, somehow caused the disease directly. In searching of the Mayan and Aztec medical texts, which there were several of, there was no note of anything that sounded like malaria, which is very interesting. However, in South America, there were mummies, as you know, one of my favorite subjects, who actually have antibodies to malaria. Interesting. Proteins are very stable, much more stable than DNA or RNA. So it is actually possible to find those proteins in mummies. And they did, antibodies that are active against malaria. So there's probably a pre-Columbian introduction of malaria into the Western Hemisphere, but exactly how that transpired has not been documented yet. But later in the 1500s, after the Columbian exchange, both European travelers to Africa and the enslaved people that they brought to the United States carried malaria probably back into the Western Hemisphere. The first major advancement in understanding the cause of malaria didn't happen until 1880. This was from an individual named Charles Louis Alphonse Laveran. He's from France, and he was working in a hospital, a military hospital in Algeria. And he investigated the blood of a person who had recently died from malaria, and he noted under the microscope that there were parasites in the blood. And this caused him to believe that this probably was the cause of malaria. In fact, he worked on this for many years. He worked on other diseases, one of which was leishmaniasis and another trypanosomiasis, but made major advancement in understanding malaria. And in fact, won Nobel Prize in 1907 for his work. That's sort of interesting time. If you recall the history here, this is the time of Louis Pasteur and Paul Ehrlich and others who were investigating germ theory as it contributes to human disease. So those were all bacterial. This was the first time that a cell, which was actually eukaryote, in fact, a protist, so a complex organism, the first time that had been found as a pathogen in a disease state. So it was a very interesting discovery. Around the same time, there was a Scottish physician named Ronald Ross, who was working in Calcutta, also in a hospital, and he was investigating the cause of malaria in birds. And he found that the birds were being bitten by mosquitoes that had these parasites in their saliva. So that was very interesting and caused him and others to start looking at the potential source of humans being mosquitoes as well. He was rewarded for his work with the Nobel Prize as well in 1902. Somehow, 5 years before Laveran got his in 1907, which I find a little mysterious, but that's what happened. After his work in Calcutta, Ross moved to the Liverpool School of Tropical Medicine and continued work in malaria. Ross was involved in the development of a number of elimination programs for countries such as Egypt, Greece, Panama during the making of the Panama Canal, and also Mauritius. And I found that one kind of interesting. Given his interest in the malaria in birds, I thought maybe he thought he could save the dodos, but they were gone by then. There have been major programs to eliminate malaria in locations all over the world ever since. And people have been relatively successful in many places, like in the US and Europe. But there have been places such as Sri Lanka, where over the course of the last 50 years, it's been eliminated 3 times and it's returned. So it's very difficult to keep it out of some areas where there are other endemic areas nearby, and it can leak back in. The first treatment for malaria occurred centuries before an understanding of what the pathogenicity was of the organism that caused it. In fact, it goes all the way back to early days of the Spanish in Peru, when they found that local people had made a tincture from the cinchona tree that they used for treating fever. Of course, it wasn't for malaria initially, but the local people and the Spanish actually tried it for malaria and found it to be quite effective. It wasn't until later that they found that the active ingredient was quinine, and the British, of course, took advantage of that in creating a medicinal drink that we know of today and enjoy as gin and tonic. which of course contains quinine in the tonic. In the same time frame, on the other side of the world in China, in 1596, Li Shishen recommended the use of a plant called Artemisia annua be used in a tea to treat malaria. Now, many people today think that probably wasn't very effective, but in looking at the use of herbal medicines and ancient Chinese remedies, a woman named Tu Yu in the 1970s with her colleagues actually investigated potential antimalarial compounds within that plant. And that led to the group of compounds referred to as artemisinins. And these are very effective. In fact, they are the primary treatment for malaria today. And those are in combination with other secondary drugs for malaria, very effective. And this, in fact, led to the Nobel Prize for her in 2015. Now I'm going to discuss diagnostics and their application in malaria. I'll go back all the way to our old friend Charles Lavernon in the late 1800s and his observation through microscopy of parasites in blood. First time people had ever reported that. And that's still the number one way in which people diagnose malaria today. Microscopy using light microscopy, using Giemsa stain, and looking at both thin and thick preps of blood smeared onto a slide, and looking for the presence of the microorganisms. So that's the way it's done today. It isn't as easy as it sounds. It takes a very well-trained microscopist to do this well. And it has problems such as the availability of the dyes, the light microscope, electricity, etc. But it is commonly used. One of its major shortcomings, besides simply those practical issues, is the fact that it's really not all that sensitive. It's typically detecting parasites right about at the edge of where symptoms occur in an infection. And there are often samples that cannot be picked up by microscopy where someone's actually infected with the parasite. And this leads to the need for better diagnostics that I'll come back to in a few moments. Now, microscopy can be used in places that have microscopy centers, which in places like sub-Saharan Africa, India, and others are in primary care clinics that have microscopists available. In some countries, that's common. In other countries, that's not at all. And so in those places, it's often the case that people use another type of assay, which is a rapid diagnostic test, or an RDT. And usually that is going to be a lateral flow assay. So typically, and this is consistent with WHO guidelines, this is going to be a test that's going to be used for a person that has a fever of unknown origin in a region endemic with malaria. It's also often used in children who are suspected to have anemia, which may be the result of having pale palms, or they've been given a hemoglobin test and they have a low concentration of hemoglobin, if that's available at that site. These lateral flow tests are typically based on the use of a particular protein found that's associated with Plasmodium falciparum, which is called the HRP2, or histidine-rich protein 2 of that particular organism. It is only found in Plasmodium falciparum, however, and that's one of the problems. There are two other problems as well. One is that that particular antigen can stick around in blood up to 5 weeks after someone has been successfully cleared of malaria. So that's a bit of a problem. And the last one is that there are mutations of that protein in falciparum that preclude these tests from working. So you actually miss people who are infected with Plasmodium falciparum, but they have a mutation in this HRP2 gene that just doesn't permit detection. By the way, there's actually an HRP3 as well, and the same problem is occurring. This typically has been the case in, for instance, South America, where there seem to be quite a few of these mutant forms of the organism, and it's now being found throughout the world, and particularly in Africa as well. So this is a significant problem. In addition to using the HRP2 antigen for these tests, it's also been possible to use two other enzymes associated with the organism. One is aldolase, the other is lactate dehydrogenase. The problem with both of those is that even though they're more commonly found in many samples, they are at a much lower concentration, much more difficult to detect. So they really don't solve the problem. There are two major ways in which people are trying to improve lateral flow assays to increase the sensitivity. One is to increase the signal. And this is typically being done with fluorescent dyes or with chemiluminescence, but those can't be seen with the naked eye. So you need to have a reader. That's a problem. Having to place a reader in the sites where this testing is being done creates new issues beyond simply the sensitivity of the test. So people like to get away from that if at all possible. Another approach is to actually use better reagents. Now, I mentioned increasing the signal, but often the problem is not just increasing signal, it's the signal to noise ratio. And what often happens as you improve signal is that you increase the noise at the same time. It doesn't improve the signal to noise, so it doesn't improve the sensitivity. But this additional approach where you decrease the noise at the same time you increase the signal makes a lot of sense. And the key approach to that has typically been to make better reagents. Better reagents being the antibodies that are used to capture the antigen from the sample. So these are things such as simply better classic monoclonal antibodies, but also things such as camelid antibodies or nanobodies or antibodies, which have been developed by a company called Alamar Biosciences that we've done work with. These are clever new ways to construct antibodies that have a better specificity, don't react with other things as well, and therefore have the potential of increasing signal to noise. So that's a particularly useful way, we think, to do that. And there are other ways to do it as well beyond antibodies, using things such as aptamers or molecular imprint polymers. There are other methods as well. Another approach that's been taken is to try to use nucleic acid-based assays instead of protein assays. That could make sense. In fact, been a lot of very good studies and publications on this using PCR and isothermal amplification methods. The problem has been, however, the cost. The lateral flow assays based on proteins are very inexpensive. In fact, they're less than $1, even sometimes as low as 50 cents. And in fact, they were by far the most used test in the world until SARS-CoV-2 came along. The number of the LFA malaria tests which are produced per year ranges between 500 and 750 million. So that's a large number. And so that can help bring the cost down. There was a lot of work in trying to use PCR and isothermal amplification for malaria testing over the last decade or so. But at that time, the technology was just too expensive. And so just couldn't be justified in a place where these LFAs are available. Now, that's changing. That's changing mostly because of SARS-CoV-2 testing. And over the course of the last 3 years, we found that people have been able to develop much less expensive nucleic acid-based simple assays that can be used in point of care or even potentially in the home. That, I think, has a potential of bringing down these costs dramatically and perhaps making it affordable to use malaria and nucleic acid-based tests in these environments. I think that we are likely to see tests which are less than $5 in the not too distant future at scale. And that may still be too expensive relative to less than $1 for the LFA test today. But perhaps in combination with other targets. Often these are being used in people that have an unknown origin of their fever, which may be malaria, often will be, but could be a number of other things as well. So would it be possible to create a small multiplex of assays of other potential fever-causing organisms in combination with Plasmodium in order to make the overall cost acceptable? We'll have to see. I just don't know, but we'll see over time. As I mentioned earlier, one of the problems with the LFA assays based on the HRP2 is that protein is only found in falciparum. Yet we know that Vivax is another significant organism in this etiology. As I mentioned, that's probably about 20% of all cases are due to Vivax infection. So that existing LFA test isn't going to work. Vivax infections are typically in places other than sub-Saharan Africa, for instance, in Asia, many places in Asia, in Central and South America, also in the Western Pacific, and then the Horn of Africa, as opposed to many other places where falciparum is typical. In a report in 2019, looking at the source of malaria infection in Brazil, 89% was found to be Vivax. There are two major problems in developing tests for Vivax detection. The first one is, unfortunately, it's at very low concentrations, lower than falciparum, which is already a problem using these standard methodologies. And we don't have HRP2 to use. The second problem is that there is a latent form of Vivax where the organism is hidden. It's in the form of what's referred to as a hypnozoite in the liver. and you just can't find any organism in the blood at that time. So that's a major problem. There's an interesting approach to detection of P. vivax that is being explored by a company called Hemex in Portland, Oregon. And this has to do with detecting the molecule that's called hemozoin, which is a paramagnetic crystal that is formed from the metabolic byproduct of hemoglobin as the pathogen actually digests it. So these crystals are characteristic of malaria. In fact, this hemozoin is actually found in all Plasmodium infections, and so is not specific to Vivax, but patients where mostly it's Vivax, which is the predominant organism, it would be very, very useful. So that's the approach they're trying to take. They've come up with a magneto-optical technology that's used on a very small instrument. They've done studies using the hemozoin detection and found very nice performance. So look forward to seeing whether that becomes available for Vivax detection. They presented the most recent results in a 2021 publication. The last type of diagnostics that I would like to talk about involves what is referred to as malaria serology. That is specifically looking for antibodies against malaria. Now, why would you want to do that? Well, there are a number of potential applications that are a bit different from what I've talked about to date. For instance, you can determine what the incidence of malaria is in a particular area over an extended period of time. You could understand whether intervention is successful, that is, efficacy of treatment. You could understand whether you've got zero transmission. You could understand whether there is a reemergence of malaria in a particular area. Because you don't have to be looking specifically at the time of the infection, the small window when the organism is detectable in the blood. This is now evidence of a previous infection or potentially a current infection. You have a broader open window of when that individual was infected. One of the other things that's being looked at with this is to try to determine whether or not there is a Vivax infection, because you're going to have antibodies even in that hypnozoite hidden state. So that could be an advantage to it as well. A few years ago, I had an opportunity to moderate a meeting on malaria serology at the Pasteur Institute in Paris on behalf of the Gates Foundation. And it was really quite an interesting meeting. I was fascinated by the investigations that a number of people were doing using, for instance, peptide protein arrays, trying to determine whether there were antibodies that had different time signatures of infection, if you will. That is, they could be used to determine whether someone had been infected over the course of the last few weeks, the last few months, or even the last few years. And the intent was to have different types of antibodies for different applications, where you want to understand whether or not there have been new recent infections in the area where you thought that had been eliminated, or whether over the course of a longer period of time, you see a difference in the number of total infections by simply look at the overall population, look at a reasonable number of people within that population and say, now I see fewer overall people with the antibodies. That probably means that the number of infections has gone down. So that's a population-based approach to this, even though you could use this to look at particular individuals like with the Vivax infection I mentioned, but most of the questions that they were trying to answer had to do with population epidemiology of the disease. So a very different way to use diagnostic tests. So there's still a lot of work going on in malaria diagnostics, and it's necessary. We have problems in things such as lack of sensitivity in the falciparum assays, the problem of the mutations that can occur in the HRP2 gene that cause these tests to actually miss cases, lack of tools for Vivax, improve the overall sensitivity of all assays, and then the possibility of using these serological assays to understand population dynamics in malaria. I mean, all these things are active areas of investigation, and we look forward to seeing the new innovations over the course of time. Thanks very much for listening to another episode of Interesting Stories in the History of Diagnostics. This is Mick Urdea for Halteris Associates, and I look forward to talking to you again soon. Bye.